So I would like to welcome everyone to the International Society for Vaccines March Global Webinar. My name is Lars Frelin from Karolinska Institutet in Sweden. I'm the chair of the Outreach and Public Engagement Committee, and I would like to shortly give an introduction to the society and then also introduce our speaker for today. So the International Society for Vaccines is a global not-for-profit organization that aims to encourage, establish, and promote the development and use of vaccines to prevent and control infectious and non-infectious diseases in animals and humans. And if you're not a member already, I would encourage you to become a member by going to our website, and you can see on this slide the benefits of being a member in our society. I also would like to highlight our annual Congress, which is in October 21st to 23rd in Seoul, South Korea. It's an annual Congress that we have every year, and I would like you to also visit our webpage for more information about the Congress this year in South Korea. I also would like to thank all our annual supporters of the society that are highlighted here on this slide, which are crucial for the operations of our society. So a big thank you to all supporters. If you are interested in what's going on within the society, you can also follow us on different social media platforms. We are on Facebook, LinkedIn, and also on YouTube. If you scan this QR code, you will come to our LinkedIn page. So I also encourage you to follow the society for the latest news, and also so that you don't miss any of our activities that we are doing. Then also I would like to highlight that already next month we have another webinar on April 24, and the speaker is Emanuele Andriano from Toscano Life Sciences, who will talk about the evolution of B-cell responses in COVID-19, mRNA vaccination and hybrid immunity. So already now, mark your calendar for this event in approximately 1 month. But the topic for today is our speaker, Dr. Trevor Smith from Inovio Pharmaceuticals, and we're very happy to have you here today. Dr. Smith, you are a molecular and cellular immunologist, which focuses your research on delivery of DNA-based drugs and against infectious diseases and oncology targets. You're the head of the discovery research team at Inovio Pharmaceuticals in San Diego, California. You have been the lead and co-author of more than 40 publications in the field of vaccine development against both infectious diseases and other targets. So we're very happy to have you here today to talk about development of DNA-launched therapeutics. So it's a pleasure to have you here, Dr. Smith, and I will hand over the word to you. And just before I do that, I would like to remind everyone that you can post questions in the Q&A function in Zoom. And you can also raise your hand by the end of this presentation, and I will moderate the session and unmute all of you that have posted questions so that you can ask the question to Trevor Smith. So thank you very much, and I hand over to you, Trevor.
Much thanks, Lars, for the kind words. And let me share my screen now. Okay, I trust you can all see the introductory slide. And
Perfect.
Excellent. All right. Well, just to start by giving a big thank you to the ISB for the invitation to present today, and also for the support they provide to vaccinologists around the world. So today I'll be giving a presentation on the development of DNA-launched therapeutics. Okay, so I do work for Inovio. We're a publicly traded company, so here's my forward-looking statements. But let's start now with the outline of today's presentation. So I'll begin with an overview of DNA medicine technology, and then I'll give a developmental update on a HPV targeting immunotherapeutic, INO-3107. I will follow that by the introduction of a novel technology, which is in vivo launch of monoclonal antibodies. This is technology we call DNA encoded monoclonal antibodies. And we'll then be providing a update on the development of a COVID-19, SARS-CoV-2 targeting DMAP. So let's start with the overview of the DNA medicines. So these are synthetic DNA medicines in which we insert our say target antigen of choice. So that could be a cancer-associated antigen, HPV associated, like HPV 16 and 18, E6 and E7 antigens, for example, or infectious diseases, including COVID, HIV, influenza, or even use this technology to launch monoclonal antibodies, which I'll be discussing later. So it's a technology in which we deliver the plasmid DNAs in vivo using in vivo electroporation, and we can use these devices to specifically perform local delivery to muscle tissue or the skin. And there's certain mechanisms involved when targeting each of these areas, for example, HPV and cancer, which we really have highlighted in many of our clinical programs. And for example, with in the HPV and cancer program, we've showed the ability to elicit cytotoxic T cells, which with these cells we aim to treat disease. In infectious diseases, we've shown the ability to drive neutralizing antibodies and T cells as well. And then technology which I'll be discussing in more depth later, instead of a vaccine approach in which you deliver obviously an antigen, we can actually code for fully functional monoclonal antibodies, and I'll show you some examples of this, how we launch these monoclonal antibodies to prevent or treat disease. And there's also the potential as well with this technology to launch a myriad of therapeutic proteins, for example, factor VIII involved hemophilia. So some of this highlighting some of the key features of Inovio's DNA medicine platform. So I mentioned before, the ability to drive both antibody and cytotoxic T cell responses. We've shown this against multiple indications. An important characteristic of this technology, if you get my pointer, is this the ability to boost. So it's just plasmid DNA, there's no anti-vector response. And it also means that these drugs are very stable. So there's really, you can negate the need for the cold chain to get the drug to the participants in need. And also, and this is the same with many nucleic acid based platforms, we can rapidly perform construct design and manufacture, again, to rapidly get these drugs to those that need them. Using this platform, we've performed more than 15,000 administrations in around about 5,000 participants. So this is a high-level overview of our pipelines. We have multiple preclinical programs, including DNA-encoded monoclonal antibodies. Technology, which I'd really like to highlight, is DNA-launched nanoparticles. So this is the technology coming out of the Wistar Institute, the laboratories of Daniel Culp and David Weiner. And it's really nanoparticles decorated with antigens, and they're very powerful tools in generating strong humoral immune responses, and they also are dose sparing as well. And we're actually testing a HIV DNA launched nanoparticle, Ino6172, in the HBTN trial currently. And including infectious diseases, we also have cancer vaccines, Ino540, BRC12 mutation, and also testing in GBM, and also 3100, which we are developing as a treatment for anal dysplasia. So this is a vaccine which expresses HPV 16 and 18, E6 and E7 antigens. We're also using that with IL-12 as a treatment for head and neck cancer. I'd like to focus now on the most advanced product, which is Ino3107. So this is our lead candidate, and it's got RRP. So what is RRP? Well, it's an acronym for recurrent respiratory papillomatosis. This is a rare disease caused by infection with HPV 6 or 11, and it's a highly debilitating disease which impacts both children and adults. So symptoms, and you see here on the left-hand side, these papillomas which grow in the larynx area here, and you see that quite significant levels of these growing in the airways here, and these can cause difficulties in both breathing and talking. Unfortunately, the current standard of care is surgery, and this means that many of these patients are undergoing multiple rounds of surgery each year. As far as prevalence, in the US, there is 14,000 active cases and an incidence of 1.8 100,000 new cases in adults. So in response to this, we have developed Ino3107. So this is a vaccine product which is composed of two plasmids. One of the plasmid encodes both HPV6, E6, and E7, so HPV6 and 11, E6 and E7, along with human interleukin IL-12 as an immune stimulator. So we've tested this in a phase 1/2 open label multi-center clinical trial. So in this trial, 32 patients have been enrolled. In these patients received 4 doses of Ino3107. So patients are dosed on day 0, on weeks 3, 6, and 9. Enrollment criteria. So patients who required at least 2 surgical interventions a year prior to day 0 have been recruited. The efficacy endpoint is the change in the number of surgical interventions in the year prior to day 0 when compared to the year following day 0, and how we count surgery. So the patients who had RRP tissue surgically removed and any surgery performed after day 0 during the dosing window, and this was counted against the efficacy endpoint. So showing the top line data from this trial, and you see here, starting on the left, how striking it is. So number of surgeries in the year prior, so patients in the year prior have a significant number of surgeries. One patient had 8 surgeries in the year prior to treatments, and many of the other surgeries, multiple 6, 5 surgeries. And then after treatment, shown in green, is the number of surgeries required in the following year. And you see here, actually, in 9 patients, so 28.1% required no surgical intervention. Overall, we saw in 81.3%, 26 patients, there was a decrease in surgical interventions a year after INO31 administration. And the median change in number of surgeries was a reduction of 3. So we believe the mechanism of action of INO37 is the induction of cytotoxic T cells. So analysis is ongoing, but current analysis showing here. So we're looking at the percentage increase pre-treatment to post-treatment of functional CD8 T cells, i.e. T cells which have activation markers after peptide stimulation and expressing granzyme A or B of PET form, so degranulation. And we show here on the right, again, the fold increase of such cells along with the activation marker. Now here on the right, this is an example of some of the impact INO3107 is having on patients. So prior to treatment, you see this significant level of papilloma in the airways. And then following INO37, you see clearance. So to give an overview of where we are now with INO3107, the phase 1/2 trial has now been completed. And the consultation with the FDA can be used to file for BLA under the accelerated approval program. I've already mentioned the significant efficacy, 81.3% of patients seeing reduction, and 28.1%, no additional surgeries was required. There's a median reduction of 3 surgeries, and we saw a favorable tolerability profile. We've received breakthrough or BTD designation in the US. I'm currently on track for accelerated approval to file in the second half of 2024. So if successful, this would be actually the first DNA therapeutic or vaccine which would be approved by the FDA. So now I would like to turn attention to a technology in which we've been developing in recent years. And this is DNA-encoded monoclonal antibodies. So this is a novel technology which enables the in vivo production of monoclonal antibodies, which are potentially a transformative approach in prevention of infectious disease and cancer. So we have obviously developed many in preclinical programs. We have been developing this technology for several years now, but more recently, we're now moving into the clinic. And we now have an ongoing phase 1 clinical trial. This is a trial supported by DARPA and led by the Wistar Institute. And this is developing an anti-SARS-CoV-2 team. So why are we developing this technology? Well, it's really end goal here will be to expand monoclonal antibody access globally. So looking at the top left here, and this is a high-level global map of where monoclonal antibody therapies are being used. And you see here, it's obviously very much skewed towards the US and Canada, 80% use there. And the rest of the world And there's obviously many areas in the developing world which were in real need of medical countermeasures against infectious disease, such as the monoclonal antibody that monoclonal antibodies could provide. That's only 20%. And this is really due to some of the complexities in and costs involved in some of the biological recombinant options currently available. So, and also the distribution of such recombinants. So us and others have developed platforms for the in vivo delivery of monoclonal antibodies. And they include DNA, so DNA delivered with electroporation, and also mRNA, so mRNA, which basically encodes for a monoclonal antibody delivered with lipid nanoparticles, and also viral vectors such as AAV, which deliver a gene encoding for an antibody. So there are potential strengths of gene-based in vivo launch platforms. They are the rapid development. Obviously, you just need the gene sequence and put it into your platform and launch it, so you can really respond very quickly to the need of a monoclonal antibody for the in a pandemic outbreak situation. The durability of expression, so it's not just delivering a bolus of a protein. Here your body is actively producing the antibody, and the time of expression, though transient, is significantly longer than what you would have with a protein recombinant. Ease of administration, there's potential here just for intramuscular administration, which is a lot more straightforward than intravenous administration. And really, again, as I mentioned at the start of this slide, the goal is to provide a bridge which we can head towards the global use of monoclonal antibody therapies. So here I'm going to give an overview of the platform of which is DMAP, the DMAP platform. So we start with identifying a antibody, and this can be say, mined out of blood samples provided by a convalescent HIV or a Ebola convalescence. You can identify the antibody, obviously sequence it, and with this sequence we can provide optimizations which are really focused on increasing the in vivo expression of the monoclonal antibody DMAP. So the sequence can then be inserted into a synthetic plasmid backbone and then formulated, and I'll discuss some of the optimizations we've made with formulations to enhance delivery. Delivery is done usually intramuscular using in vivo electroporation. So this facilitates the delivery of plasmid DNA into the myocyte, and then the myocyte then becomes the biological factory, and then produces and then secretes into circulation functional monoclonal antibodies. Let me give you an example of this technology in action. So taking a prototype DMAP, you deliver it into the mouse, into the leg muscle of the mouse, and then you can measure expression of which may be a human IgG. So looking at the site of delivery, so this is the local muscle tissue here. This is no treatments, control treatments, and we're looking here at human IgG expression. And here in green, you're seeing expression of the human IgG, and this is in the cross-section of the muscle tissue, really showing that the majority of cells here are producing the antibody. This is perpendicular, and here is going parallel across the mushroom tissue. So really strong expression. I'll provide some more examples of this later. And then we can look, see if it's basically the muscle cells are secreting the antibody into circulation. So look at serum levels of the DMAb, and it's shown here in blue. So you see here, this is compared to the recombinant counterpart. So quite different PKs here. Obviously, a bolus injection, you get immediate levels, which are detectable in the periphery, but then they decline. Whereas here, slightly slower accumulation of the antibody in the periphery, but these levels are sustained, and we really do get durable expression of our antibody. So then we ask the question, are these in vivo produced antibodies, do they function the same as their recombinant counterpart? And the answer is yes. So this is looking at antigen binding between, again, red recombinant versus the DMAb counterpart. And we've also done this in other models such as virus neutralization and challenge. So together with the Wistar Institute, we've developed many monoclonal antibodies covering a myriad of disease targets. So on the right-hand side, you are really focusing on the infectious disease targets, so through influenza, Ebola, dengue, chikungunya, and SARS-CoV-2. So the SARS-CoV-2 is a target which we'll be covering later, and it's actually undergoing clinical evaluation. We've used different approaches, so like the SCFV approach, and we've shown that that's effective in models of RSV. And against also bacterial targets, we've shown that these monoclonal DMAbs can be effective in protecting against disease in multiple models. Also in for cancer indications, we've also shown proof of concept. So my group has been focused on some of the ways in which you can optimize delivery of the plasmid DNA, thus enhancing gene expression, enhancing expression of the transgene for the monoclonal antibody. And this has been a key optimization in getting this technology to work. So let me give you a brief overview of in vivo electroporation, the mechanism involved. So you would start by generally, for DMAbs, this is going to be the muscle tissue. You would inject, say, the DNA vaccine or the DMAb formulation into the tissue. And then at the site of injection, you would perform the in vivo electroporation. So this is short electrical pulses, which cause the temporary openings of the cell membrane. And this allows for the efficient passage of the DNA into the cell. And then once in the cell, then the DNA can then hijack the cell's machinery, and the cells can then start producing the encoded protein. We've shown efficient delivery targeting different tissues. So intramuscular, we have a commercially ready intramuscular device, which is the 5PSP, which we're using to deliver the INO-3107 RRP immunotherapy. And we also have an intradermal device as well, which is the 3PSP, which is a small handheld device, battery powered, very amenable to use for infectious disease targets. So really easy to transport, easy, not a big footprint, and very tolerable, as we're really only targeting the layers of the So showing here examples of electroporation and how it does enhance gene delivery. So we're showing GFP expression, a GFP transgene. So when you inject a GFP transgene with LTP, you really get very little, if anything, signals. These are cross sections of rabbit muscle. However, if you inject the GFP and then perform electroporation, you get a significant uptake of the DNA and production of the green transgene. And another important optimization was the combination of electroporation with enhanced formulation. So these enhanced formulations are generally enzymes, and these enzymes temporarily disrupt the extracellular matrix of this tissues and muscle tissue. So these can be used, for example, like Hylenix on label to enhance the delivery of our DNA. And we're showing here, so this is the signal as RFP, in this case, EP plus enhanced formulation, giving a stronger signal than just EP by itself. So combining Selectra delivery with formulation, we're getting gene expression levels of over approaching improvements of over 1,000-fold. And another optimization which has taken place in recent years is making sure that the fluid distribution is matched with the electrical field. So co-optimization of both of these parameters is really important for the most efficient delivery of our DNA medicines. So the DNA and E-field distributions are aligned. And we're seeing here, we get this really nice uniform gene expression across the tissue. Again, it's rabbit tissue showing that all these cross sections, you're seeing good expression of your transgene. So we've applied this technology in multiple models now. Highlighting here, which is maybe one of the most translatable, translationally relevant models. That is the sheep. So this is a model which was developed by Kevin Holvert's group at the University of Leuven. They developed an ovine, a species-matched anti-human CE monoclonal antibody, which is then put on a DMAP-like platform. And we've shown, using our delivery parameters of this monoclonal antibody, you can get robust expression in these animals. And these are big animals. We're talking here, I think this one was up to almost 60 kg, so it's the size of a small human. You're getting robust expression, which I mentioned again, it is durable expression. We've also further tested this group, this model using clinically relevant dosing levels. So these are really actually quite low dosing levels. So going down to 0.025 mg per kg and 0.1 mg per kg, we're still getting decent expression, decent durable expression. And importantly, at these levels, we're not seeing any anti-drug antibody response. So now I'm going to provide an update on a program which we've been developing under the support of DARPA and led by the Wistar Institute, and that's the development of SARS-CoV-2 DMAPs. So the project's goal here is the development of immediate medical countermeasures as pre-exposure prophylactics, and these have the potential to provide long-term protection against COVID-19. And our approach has been to use the improved delivery methods and identified formulations, along with sequence optimizations, to design and then develop DMAbs, which have been based on human COVID-19 monoclonal antibodies, which were isolated by the James Crow group at Vanderbilt. So these antibodies, which were isolated, were very potent, especially when you formulate them in a cocktail. So it's two antibodies. One is AZD1061, which targets a class 3 site on the RBD of SARS-CoV-2. And it works in unison with AZD8895, which binds to a separate site on class 1 RBD. And these were developed by AstraZeneca as the products Evusheld, the combination monoclonal antibody product. So they showed that this is a long-acting cocktail. They include the extended half-life mutation, which is the YTE, which increases the in vivo durability. And also was developed early in the SARS-CoV-2 pandemic. It did include the TM, which is a modified FC, which is the aim to reduce the FC gamma binding to FC gamma receptors, thus reducing any potential for ADEs. And Evusheld, or AZD742 cocktail, was approved as a prophylactic against COVID-19. So based on these parent antibodies, we designed DMAb versions. So we performed optimizations, which include nucleotide and mRNA structural modifications for enhanced expression in vivo. Other modifications included, we reverted back to the wild type IgG1 FC, as during the time of development of these DMAbs, it became understood that ADE was not a concern for SARS-CoV-2. And the antibodies were developed as a cocktail product, and as a total of 4 plasmids, each plasmid encoding for either a light chain or a heavy chain of that clone. So showing an example of these DMAbs in action. So part of the preclinical development program, we did testing in multiple models. And one of these models was the hamster model. So the hamster became really the gold standard model, small animal model, to test SARS-CoV-2 medical countermeasures, as it could be infected with the wild type virus and display some of the symptoms of COVID-19 display. So in this study, we tested the wild type against the TM DMAb cocktail, and we had a naive control. On day 0, the hamsters received the antibody cocktail, and we measured expression throughout the duration of the study. And you show here, so this is the level of DMAb detected in the serum. And you see here significant levels were circulating in both the treated groups. Were these antibodies functional? The answer was yes. This is against wild type virus, showing neutralizing. Here we're demonstrating neutralizing activity. On day 18, the animals were challenged with the virus, and we monitored disease. So on day 22, we assessed the lung tissue. So you see high viral loads in the non-treated animals, but significantly attenuated levels of virus, very low levels of virus in the DMAb treated animals. And Also, this is in the nasal tissue, which is actually a more stringent measure of protection. Again, you're showing significantly reduced levels in the nasal tissue. So this is very encouraging data. This and other data showed that we were getting good biodistribution of the produced antibody into the tissues associated with disease. And also, we saw a positive impact, and that there was really minimal, if any, weight change in the treated animals, where there was significant weight loss in the control group. So as part of the non-clinical development program, we look to translate the technology to the rhesus macaque model, which again is a model which can be naturally infected with SARS-CoV-2. So in this model, we tested 3 separate groups. We had a control group, and then in group 2 and group 3, we tested clinically relevant dosing. So this is both the level of dosing and using the clinically appropriate delivery protocol, which is IM administration using our Selectra system. So we group 2 had the DMAb with the YTE, and we tested that against the DMAb with the wild type DMAb, so without YTE. So showing here, first thing we did, so day DMAb administration on day 14, we measured DMAB expression in the serum. And you see here, different levels in the microgram per mil range in the rhesus macaques. So we also wanted to determine whether the expressed antibody was functional. So we performed here neutralization assays against wild type live virus. We're showing activity in both treated groups on day 0 and day 7 here, and also against pseudovirus, wild type pseudovirus in the treated groups. And we're showing against the Delta variants of concern, live virus, and pseudovirus. And importantly, with this antibody cocktail, there was one of the few antibody cocktails who actually retained activity against Omicron. So confirming here, even though the levels were attenuated compared to the earlier wild type pseudovirus variant, pseudovirus, you still did get activity against the BA2 Omicron. So on day 0, we challenged the animals with 2.8 times 10 to the 6 TCID50 of the Washington version of the virus. So this is a very stringent challenge model. Many of the other challenge models were using lower levels, 10 to the 5, 10 to the 4 level. And also this challenge model included intratracheal, oral, nasal, and ocular administration of the virus. And so we monitored clinical scores, and you see here, so the monkeys do develop a reasonably mild level of COVID-19, but it's still detectable through clinical scoring. And you're seeing here, these are the clinical levels peaking on day 3. And then in the treated groups, you can clearly see the attenuation of the clinical scores in both groups. So as far as the pathology, we performed necropsy scoring in the lung tissue, and again showing here, so a significant percentage of the total lung involvement in the control group, but a significant reduction in the group 2 in this necropsy score, and also trends in reduction in group 3. And as far as pneumonia score, we also noted significant reduction in both the treated groups. And then looking at the H&E stains, you see a strong stain in the control group, but again, attenuated levels of cell infiltration in the DMAP treated groups. So in summary, the SARS-CoV-2 DMAP cocktail demonstrated durable expression. It demonstrated activity against multiple SARS-CoV-2 VOCs, including Omicron, and protected against disease in multiple animal models. As a program update, the data presented was used to file an IND, and a phase 1 clinical trial has now been open. And this is being run by Pablo Tebas at the University of Pennsylvania, which is a dose escalation study evaluating safety, tolerability, and PK profile of the cocktail using the optimized delivery protocol. So I'd like to finish, most importantly, my talk by thanking the team involved in this work. The SARS-CoV-2 DMAP project was led by David Weiner and included very important contributions from Ami Patel's group and Dan Culp's group, the group here at Inovio, the clinical trial run by Pablo Tebas's group, Mark Esser and Paul Leon, especially at AstraZeneca, Jesper, who at the time was at Indiana University, who's now at Wistar, and also for access to challenge models, Dom Cabasa, and also the team at Rocky Mountain Laboratories as well. Thanks for their work with the resistant cat challenge model. And also support from DARPA, and also support from the Bill and Melinda Gates Foundation. And these two organizations provided funding across multiple of the DMAP programs. So with this, I will take questions.
Thank you very much, Dr. Smith, for a very nice presentation. And now this talk is open for discussion. We have a few questions in the chat, and I also would like to see if anyone would like to ask a question. Also, you can raise your hand. But I think we can start with one question from one of the participants, and I would like to ask if A. Rosenberg would like to ask your question. I can unmute you so that you can ask the question that you have.
Sure, thank you very much. That was great. So my question has to do with the cell types that are the production sources of the vaccine antigens or the monoclonals. So are the vaccine producing cells, whether they be muscle or myeloid cells that also take up the construct, are they destroyed by the elicited immune response? And as well, is muscle function impacted by the transduction and expression of the transgene? I mean, the liver, liver cells are good at producing proteins, but muscle cells produce a very restricted protein spectrum. And so the question is whether their function is impacted.
Yeah, so that's a very good question. And no, we haven't seen any impact in function, and say this is transient expression. And what we do find that the muscle cells are a very appropriate cell for this. And as I say, they're multinucleated and they're highly secretory, so they can express and secrete functional we've done this with monoclonal antibodies, other therapeutics like DNA encoded bispecifics, ones which target CD3 and a cancer antigen. And we can get, as I say, it's long-term expression, which we've seen. So that really, and it's probably, there's obviously different mechanisms involved with if you're launching a cell protein, so something so like the DMAbs, which are human monoclonal antibodies that you aren't inducing a immune response against the cells. Though of course in vaccine studies we do see a infiltration of immune cells into the site. But as far as muscle function is concerned, I say we've tested this in 5,000 plus patients now, and yeah, really not a negative impact on the what we can see is muscle function.
Thank you.
And then we have 2 hands raised right now, and I think we start with Jane, if you would like to ask your question.
Thank you. If you can hear me, it's really impressive technology. I was kind of thinking about the DMAb approach. So as far as I understand, it doesn't really have a kind of an off switch or anything that you could use to intervene once you've kickstarted it. So are there kind of safety implications, and if you're able to share, did the regulators comment on any aspects of that? Thank you.
Yeah. So yeah, you're right. We don't currently have a off switch, and that's some of the reason which goes and why we are targeting. So the first clinical trials have been done with targeting infectious disease. Okay, so these are human antibodies which are being produced, which were actually matched to those, what do you call this, mined from human convalescence, obviously against a foreign target. So obviously, and we performed tissue cross-reactivity studies, and there was no cross-reactivity of the product with human tissue. Obviously, that's a different situation for if we're doing other therapeutics. For example, if you're developing therapeutics targeting the checkpoints, so the anti-CTLA-4s or anti-PD-1s, which is then potentially the need for off switches or on switches to control expression. So if you do then see obviously a safety signal, you can turn expression off. So that is something which the field is working on, and multiple people across the field are working on developing these off switches. So yeah, it's definitely a consideration. Though for say the approach we've taken first with the monoclonal antibody targeting infectious disease, the requirement for those switches is really not needed.
No, thank you. I was also thinking about anti-idiotypic type of anti-drug antibodies that you might get even with your infectious agents. But yeah, thank you.
So what type of response did you say?
Anti-idiotypic,
right.
even if it's
Yeah, which we haven't seen, as say we don't see measuring ADAs against the anti, and we really haven't seen that when you at least if you're species matching the launched antibody into the animal, and the level of ADAs. I mean, you sometimes do detect it, but it's similar to what you see with recombinant monoclonal antibodies.
Very good. Thank you. Thank you so much.
Then we go to the next person that would like to ask a question, Warren.
Hi, can you hear me?
I can.
Hi. Yeah, I just wanted to ask about like delivery of DNA therapeutics to cancer tumors and stuff. Something I've been looking into is adeno-associated virus delivery of like I'm working on a self-amplifying DNA system, which essentially might provide an off switch in that it's like virally derived, and it seems to trigger like a long-term expression, but then disrupt the cell's ability to replicate and potentially activate DNA damage response mechanisms. Yeah, so I was just wanted to hear your thoughts on whether or not like with delivery to intratumoral cancer tumors, can you like, to what extent can you limit the spread of the DNA to, you know, if you have some sort of suicidal DNA that can express an anti-PD-L1 or some other cytokine-based therapy to the microtumor environment that is itself suicidal. Do you think that that would be better suited to limit the spread of the therapy to only the target cells, in that the suicidal DNA might it would potentially kill any cell that it takes it in, so it kind of limits the amount that it can be expressed, but in the same time activate DNA damage response mechanisms, which might enhance have an adjuvanting effect for CD8 cells.
I could see the advantage potentially of such technology, especially in driving productive immune responses. I'm not wholly aware of this technology, but I'd love to do more reading and talk to you further about this technology. I mean, currently this is what we do with plasmid DNA. It is local delivery, so you don't get great biodistribution of the plasmid DNA, and it really not, or very, very low levels going into cells outside of the injection site, because what you need is the local electrical fields produced by the EP to get it into the cells. So you're not getting really, say, the vast majority of expression is in the local tissue. You could see with technology you're talking about, into tumor tissue, there's something which I haven't done, the delivery into tumor tissue. I know other companies have done delivery into tumor tissues. IL-12 has been plasmid expression. IL-12 has been delivered into, I believe it's melanoma tumor tissue. The mechanism you're talking about is interesting, and maybe similar to mechanisms operating in the skin. We do vaccination to the skin, in which we do see quite a high level influx of immune cells into the site, as the skin's generally more immunocompetent, I would say, than the muscle. There's more immune cells in there, which we see actually a dose-sparing effect generally going into the skin. I can't really comment much more about that.
Thank you. And then we have a last question from Dr. Sinha. So a quick question and a quick answer.
Am I audible now?
Yes.
Yeah.
Yes. So my question is, as we understand that after injection of the DNA, slowly the translation level will go down. So what do you think should be the boosting strategy, and that will In on one of your slides, I have seen that the boosting strategy is 6, 7, 6, 9 weeks. So what should be the boosting strategy? Seeing the serum concentration, or whether it will trigger the ADA after boosting? So what are your experience about that?
Okay, yeah. So the boosting strategy, that was for a vaccine like product. Okay, so with that, we've performed. I mean, it's obviously a case-by-case basis on what the most appropriate schedule for that is. And then I think you're also referring to the DMAb technology, and obviously it's very different what we're delivering. We're delivering human monoclonal antibodies, so we're not actually raising an immune response against these antibodies. So redosing is possible. We've done that in preclinical models to show that there's the potential to redose without eliciting an ADA.
So when you talk about formulation, so as this formulation is related to the, I would say, safety of the DNA, so that the DNA does not get degraded, or any other terms you were telling about formulation?
Okay, so the formulation, and again, I'm supposing that you are talking about the DMAb formulation, so the use of enzymes. Obviously, enzymes are selected which don't degrade the DNA, and make sure the DNA is stable in that formulation. And yeah, and so they used actually on label. So the product which we use to in the formulation is approved for intramuscular administration, and it doesn't have any negative impact on the stability of the DNA.
Thank you.
So that will conclude this webinar today. So we thank Dr. Trevor Smith very much for an excellent presentation. It's also a nice discussion. So thank you all, and I hope to see you in 1 month again at the next webinar. So have a good day and see you later.
Thank you.
Bye.